So let's look at an experiment where they did a test without any control group and then what they did to fix it. So this is an experiment with what we call gastric freezing. What they did is, in the late 1950s, they were trying to treat stomach ulcers. And so the original experiment was with 24 individuals, and they lowered a balloon down into the stomach that was filled with refrigerated fluid. And thing is that the argument is that by using this supercooling, acid production would be reduced and the ulcer would be relieved. And in the experiment, then in the 1950s, is that, yep, 100% of the patients experienced relief, and therefore condition improved, and this treatment was used for many years. However, a few years later, another researcher decided to do the experiment with a more careful design. What they did, you'll see first, they used more subjects and then divided the subjects into two groups. And one group got the treatment, so they got the gastric freezing with the refrigerated fluid, and the other group got a balloon inserted with a room temperature fluid. So this guy right here is the control, right? Because room temperature is not far off from body temperature. It's a little cooler, but not much. And they're still getting a balloon inserted, right? So they've got the same treatment for everything else. The only difference is the actual treatment, the actual variable, the treatment variable, which is the gastric freezing. And when the experiment was run, what they found is actually a little bit more of the control group experienced relief compared to the gastric freezing group. And so due to this experiment, gastric freezing was abandoned as a treatment for gastric ulcers. So control groups are incredibly important for experiments because what they do is they allow us to eliminate the placebo effect. This is the phenomenon in which people respond favorably to any treatment. And this happens because of the power of our mind. Okay? The thing is that our mind is pretty powerful, and if we think we are getting a treatment, sometimes it actually can work. So some people, if they believe they are getting a medication to treat a certain condition, it sometimes can kind of trick their body into kicking in, an immune response or something like that and get it to work. Sometimes it's just, pure happenstance that they get into the trial, they're on the placebo group, and their body goes into whatever response it is that they're supposed to be going into. And so this effect is why we need this control group. The thing is, with that gastric freezing experiment, all of those patients knew they were getting the treatment, knew, hoped and thought that it should give them relief, and so it did. The thing is, when you've got control groups and nobody knows which one they're getting, the placebo effect can still occur, but the control group controls for that because the placebo effect will happen equally in either group. And so that will help you eliminate that placebo effect out of the two groups. So how can you help eliminate more of this placebo effect? Well, when you design your experiment, first have a control group, but second, you can design your experiment using what we call blind design. So blind experimental design First, you don't let your experimental subjects know which treatment they're receiving, right? So whether they are in one of the treatment groups or if they are one of the placebo groups, right? So if they're getting the sugar pill or whatever it might be, they don't know. So the subjects have no idea in just a straight blind experiment, but the researchers do. So the researchers know which of the subjects are getting which treatment, which for some studies is okay, but there's even a better design, and that is a double-blind experiment. We'll talk about why a double-blind experiment is better here in a second. Because here, neither the subjects nor the experimenter knows which treatment the subjects are receiving. So you might be like, well, then how do they know what to give anybody? Everything's coded. So it's a computer code for which one's which. So that way, at least somebody knows who's supposed to be getting which medication, but the researchers, the experimenters don't know. Because there's an issue with knowing which subject is which. And the last thing to look at is making sure that your subjects are randomized. You want to make sure that they are assigned into the different experimental groups randomly so that there's no influence on the composition of the groups. You want to make sure that you're not unconsciously putting specific individuals into the different experimental groups because you think that they would be better in one group over the other. So if you have a program, randomize them so that you don't have any choice into it. That's the best option. So why? Why is a randomized, double-blind, controlled experiment the best option? Well, if you've randomized your subjects and it's double-blind and you've controlled all the variables except the treatment, that means whatever your results are most likely, as much as you can, a result of the treatment and not anything else. And so why do I recommend the double-blind experiment? The thing is that we have biases, and we have biases that we don't notice all of the time. So they did a study on publishing papers in a journal called Behavioral Ecology. They didn't do it on purpose. What they did is they changed the policy for reviewing manuscripts. So before they changed their policy, the reviewers knew the names of the authors, which means they could assume the sex of the author, right? After the policy was in effect, the new policy, which was double-blind, that meant that they didn't know the names of the authors. So the reviewers didn't know the names of the authors, which means they could make no assumption on the sex of the author. What they found is, before the new policy, only just about 24% of the papers that were published had a female first author, meaning the first name on the paper was a female. When the new policy was enacted, that number went up to over 31%, which was statistically significant when they looked at it. And that meant that unintentionally or intentionally, the reviewers were selecting papers with male authors as the first author to be published. Once they didn't know who the author was, they published more of the papers with a female author as the first author. So if you don't know your subject's, name, sex, condition, whatever it is, putting them into the double-blind study and not knowing whether they're getting a placebo or a treatment actually keeps you from being able to enact your biases, whether they are, on purpose or not. The last thing about experiments is repeating them. It's incredibly important to repeat experiments, so replication is just the process of repeating an experiment. So why would we want to repeat an experiment? What do you think? So repeating an experiment increases our confidence in those results. If we can repeat an experiment once, twice, 3 times, and we get the same results, we're pretty confident in what happened there. It also helps isolate variables responsible for outcomes in experiments, especially if these experiments are repeated by different researchers, because then we can say, oh, well, this is a slightly different outcome, so it may be because if we're working with people due to population differences, something like that. So experiments that can be repeated again and again and again are incredibly effective defense against biases, because if they're repeated, and like I said, by other researchers, it removes those biases, or can help remove those biases because it's different individuals working on them.